Good afternoon, everybody. My name is Anna Vangone, and I'm postdoc at Utrecht University, as I said. And so first, I really would like to thank the organizer to give me the opportunity to speak here today. The 3DSIG has been a great source of inspiration for me since 2011, the first time I joined. So it's really a pleasure for me to be here. Today, the topic of my talk is going to be prediction of binding affinity in biomolecular complexes, with a particular focus to protein-protein interactions. So as many people in the audience might be familiar with, protein-protein interactions are at the basis of almost every function that happens in the cell. So if we wanted to study a particular function, it's very important to study the complex or the complexes that are related to this function. And we can study those complexes in many different ways. It's very interesting to know the structure, for example, but what I am going to talk today, it's the binding affinity. Then, in other words, is the energy that keeps the two proteins together. The computational prediction of the binding affinity, so this free energy of the association process that you see here, has been a long-standing challenge with more than a couple of decades of active research. Now, in the computational field, we have several approaches that allow us to do an estimation of an energy that, in a very general perspective, we can group in two main families. So we have the exact method, think, for example, molecular dynamic based, that they can provide an estimation of the energy, but they cannot actually be efficiently applied for such big systems as proteins. On the other side, we have the empirical scoring functions, for example, scoring in docking, that provide a rough and fast estimation of the energy of a system. But it has been shown that so far, none of those approaches can actually reliably predict the binding affinity. Now, considering the limitation of energy-based method regarding the prediction of the binding affinity, we decided to tackle this challenge from a different perspective. And that is why today I'm going to talk about a structural-based approach to predict the binding affinity. So, there are several structural properties of a protein-protein complexes that have been looking at. For example, the buried surface area, that is the surface that is buried upon complex formation. So the first time, so this is the first structural properties that has been introduced to describe the binding affinity, and it's also a very broadly structural property used generally in the topic of protein-protein interactions. So it has been shown that in some cases, the buried surface area correlates with the binding affinity. So the broader is this buried surface area, the stronger is the binding. But also this property, it actually shows limitation in terms of accuracy. A step forward was done a couple of years ago, when a PhD student in the group in which I am hosted now realized that not only the properties of the surface, but also the properties of the non-interacting surface, so this part that you see here in red, they both overall contribute to describe, and in other words, to predict the binding affinity. So that was a step forward to the approach merely based on the buried surface area, but still showing some limitation. What I introduce as a novelty in this field is looking at the interfaces in terms of contact. And I'm going to show you what I mean. So as contact at interface, I simply mean the number of residues that are within a certain distance threshold. The results that I'm going to show you today correspond to the threshold of 5.5 angstrom, but this threshold has been optimized with several iterations and experiments. So in this little example that it's reported here, we can see, for example, that there are 6 possible interface contacts. So it's a very simple definition. So we can count 6 number of interfacial contacts. But those contacts, I also wanted to classify them according to properties of the interacting residues. So for example, looking at the polarity or looking at the hydrophobicity of the interacting residues. And I'm going to show only the results related to the classification based on the polarity, so polar, apolar, and charged residues. So in this example, we will have different features that correspond to the different number of contacts according to the different classifications. So for example, the property 1 with the number of contacts between charged and polar residues, and so on. So since I have all those features that are the different number of contacts at the interface, I tried to train a linear regression model. So very simply, I applied a linear regression model, so for which each of these properties I attributed different weights. I also applied a feature selection method to apply overfitting when you have only a few test cases, and I also cross-validated the results. Of course, I also used a training data set. So I used a protein-protein binding affinity benchmark for which both the binding affinity and the structure have been experimentally determined. And this binding affinity benchmark, it's a very heterogeneous data set. So it has a lot of different functional classes, different values of delta G's, of barrier surface area. So I have very tiny complexes, but also very big ones. So it's quite heterogeneous. So now some results. So by checking, the features that I analyze are this number of contacts, and I use this data set to check what's going on there. So in this plot, you can see the first results in which I reported the number of contacts reported on the y-axis as a function of the experimental binding affinity. So the first thing that I observed is that the more is this number of contacts, the stronger is the binding. So there is a kind of correlation between those number of contacts and the binding strength. This correlation resulted to be of minus 0.5. Interesting also is to look at the correlation that the barrier surface area showed for this very same complexes. So each of these dots is a complex of the benchmark. And the barrier surface area showed instead a lower correlation of minus 0.3. So it was interesting already to see that the contact correlated with the binding affinity and correlate even more than what is so far the standard structural property that is used in the field. Another interesting aspect was when we looked at those complexes by sorting them according to the experimental method that was used to measure the binding affinity. In particular, when we saw the data that were measured with inhibition assay and fluorescence, we saw no correlation at all. So we decided to keep going our analysis, but removing those data, so keep going with the other one for which correlation was at least minus 0.5. So we removed those two sets of data, and we kept going with those complexes. So based on this observation, I tried to train a linear regression model. So the first one, very simply, if we just train a predictor for binding affinity based on the total number of contacts, the correlation between the predicted binding affinities and the experimental one resulted to be of 0.59. But that's also how I wanted to train a regression model in which I use as a feature the contact but property-based, so classified by different properties. And so by giving more weight to some contact and less weight to others, the final predictor was resulted to have a correlation of 0.67. And finally, I decided also to add this non-interacting surface, because it has been shown in this work, previous than mine, that also this surrounding part might contribute to the description of the binding affinity. So I trained a model that, as you can see here, has both some of those contact at interface and also some of those properties of the non-interacting surface. And this predictor resulted to have a correlation of 0.73. You can see here the predicted delta G's versus the experimental one, so it's a nice scatter plot. Those data have been, of course, cross-validated, so those results and also the weight of the predictor are quite stable for also after the fourfold cross-validation. Of course, I wanted to compare how this approach is sitting in with the current developed approach. So the approach developed so far. And you can see here the performance of my contact-based predictor. Here is the predictor that include the non-interacting surface, and this is the Burr surface area based one. All the others, they come from the char PPI web server that include 105 scoring functions. So this approach, this simple structural-based approach, is actually showing the best performance in predicting the binding affinity in respect to these 105 scoring functions. Not only, but it's also very stable when we divided the complexes based on the flexibility that they show upon binding. So in some cases, as for example in docking, flexible complexes can be very challenging. And as flexible, I mean the one that undergo a lot of conformational changes between the unbound structures and the bound. So this approach seems also to be very stable for both flexible and rigid complexes. Then we try also to get an idea why, how come that this contact might be more informative somehow, or at least they correlate better with the binding affinity instead of the Burr surface area. And you can see here in this plot, I reported in pink, so in graduation of pink, the contact that are at the surface, at the interface, according to the contact definition or to the Burr surface area definition. And the pinker is the residue, means that that residue is more involved in the final quantity in describing either the contact or the Burr surface area. So the first thing that you can observe here is that the surface is basically the same. So the patch is mostly the same. So the difference doesn't come from the fact that we can get more residues with one definition of another. We get more or less the same residues. But the difference, and you can see from the different colors, is that it seems that when we try to describe the interface in terms of contact, these take more into account the location and the geometry, let's say the topology of the surface itself. Because we can see here that some of those residues, they might be in a very promising position, they gave more contact. While if we look here, the color is flatter. So those residues are all buried at the same way in some aspect. So it seems that by describing the interface in terms of contact, it's a kind of a higher resolution definition of the surface that we can give. Now, considering the nice performance of this predictor, we developed it in a web server that we called PRODIGY. PRODIGY is freely available online at this address and is also in a GitHub repository, so you can install it locally. I will make the slides online available, so you can also check the links. So it's really fast. In a couple of seconds, you can put your complex in and you get your answer back. Now, before concluding, I would like to very briefly show you one of the work in progress that we have so far. So we decided to apply this approach also to other biochemical molecules and to see whether these can be, those concepts can also be applied for other complexes. And in particular, I'm going to show you here some preliminary results that we get for protein ligand complexes for what we are developing and calling PRODIGY League. Of course, we changed a bit the protocol, so the data set is of course different because we need protein ligand complexes with known binding affinities. So we used complexes from the 2P2I data set. And we also changed the structural features because in this case, we are analyzing atomic contact. The ligand is very tiny, so we needed a more refined definition of the interface. So we are using so far atomic contact. We define a 10.5 angstrom. Again, this has been a threshold that has been at least so far optimized. And also we use a very simple classification so far in which we classify those atomic contact based on the atoms involved, so oxygen, nitrogen, carbon, and others. And we also added a few energetic features that we calculated with the ad hoc server. So we added the electrostatic interactions, van der Waals, and desorbation energy. All those features, so those atomic contact and those energetics, we throw in a linear regression predictor and with the feature selection. And the nice thing is that we applied this initial predictor that we have to D3R. D3R, similarly to CASP and CAPRI, is releasing basically protein ligand complexes for which, a target made of a protein and a ligand for which, among the other things, such as the poses, you also have to predict the binding affinity. So last winter, the Grand Challenge 2 of D3R was released, in which they provide the FXR receptor and 102 ligands that were binding to this receptor. So we had to predict the binding affinity for all of those ligands and rank them according to the affinity. And this is the performance of PRODIGY League, sorry. So we were quite happy about these results. First of all, because these are preliminary results. Those are very preliminary, this is a very preliminary predictor that will be developed, and we really believe that there is room for a lot of improvement. There are many things we can still try. And also because very often in a protein ligand binding affinity predictor is either target specific or ligand based, while PRODIGY League in this aspect will eventually provide a very general approach to predict binding affinity. So hopefully we can push it and get in even better than this. So as a conclusion, as a take-home message, what I show you today is that the number of contacts at the interface are a simple but robust predictor of the binding affinity, and they can also be used eventually to describe interfaces because there seems to be a kind of a higher definition parameter of to describe interfaces than what is commonly used as Borel surface area at the moment. We have also a web server that is PRODIGY. And there seems to be promising applications with the same concept also for other systems. Look, for example, PRODIGY Ligand. The use of contact at the interface has been used in this work for the first time related to the binding affinity, but is not the only application. It has also been used, for example, for scoring. So if you wanted to know more about the application of contact, you can check the poster of Romina, poster number 36. So finally, I would like to thank a few people that worked with me. So definitely Alexander Bomban, that always supported me and believed in whatever I want to have to work so far. I wanted to thank all the colleagues that were that helped me in developing the PRODIGY web server. I also would like to thank Nevia Citro, that was a master student visiting our lab, that was working in some of the part of PRODIGY Ligand, as well as Anna Marabotti, which is here, and she has also a nice poster. And of course, thanks to my funding, and to the group that I have in Utrecht, the CSB group, including Marina, that is a master student that was in our group, and she is also here with a poster. Thank you.
Time for one or two questions. Rafi has a question.
PRODIGY League, is this I guess it's the source of what's in the poster on 838. So I don't know if the question is pertinent now, but there are plenty of much larger data sets of protein ligand KD or other measures of affinity. Have you tried with a larger data set to cross-validate?
Not yet, but this is not yet, but this is something definitely that we are going to try. We wanted to make everything more robust, so definitely. No, the poster of Anna Marabotti is about something else.
In the protein-protein work, you showed, actually when you were training, you showed some experimental results for binding affinities.
Yes.
And you threw parts of the experimental data away. Yes. And those were quite different. I mean, what's the justification for that? So
the thing is, what I believe, and also I try to get some feedback from experimental colleagues, is not that those techniques are not suitable to predict binding affinities, but are not suitable to probably predict the absolute values of binding affinity. Things about, for example, inhibition assay. That is a perfect technique if we wanted to have a relative binding affinity between a wild type and mutant. You run all of them together, you can have a perfect relative binding affinity. So for mutagenesis, it's perfect. But probably those techniques are not accurate enough to give you an information about the absolute binding affinity of the complex. So probably this is the reason why those two techniques were so different by all the others. So suitable, but maybe for a relative binding affinity. So probably this is the idea we have.
Okay, thank you. All right, thanks very much. Let's thank our speaker again.
Thank you.